Good morning students. Today we are going to discuss regarding the metabolism of sulfur containing amino acids. So we have already finished with the metabolism of branch chain amino acid and aromatic amino acid. So I hope you remember what are the various sulfur containing amino acid, methionine and cysteine. These are the sulfur containing amino acid, out of which methionine is essential while cysteine is non-essential, as it can be synthesized in the body with the help of methionine. Now, apart from being an important source of sulfur in the body, these are important for methionine plays an important role in initiation of protein synthesis, as well as it acts as an lipotropic factor. And the most important thing why we are discussing them is because disorder in their metabolism can lead to various disorder, which includes homocystinurias as well as cystinurias. So that is the important thing you have to remember, the disorders which will be related to the sulfur containing amino acids. Now first of all, coming to methionine. So methionine is a glucogenic amino acid. Okay, it is a glucogenic amino acid which produces succinyl-CoA as its product, and there are three important roles of this methionine. It will be utilized for the synthesis of active methionine in the form of S-adenosine methionine. It will be synthesizing homocysteine, as well as I told you, it will be utilized for the synthesis of the other sulfur containing amino acid, cysteine. So first of all, about the synthesis of SAM, or the active form of methionine. So the enzyme required here is S-adenosine methionine transferase. This is the enzyme which is involved, and here ATP will be involved. The important point here is that all the three high energy phosphate bonds, they will be utilized for the synthesis of this S-adenosine homocysteine. So as such, it does not contain any high energy bond, the phosphoryl group, but this is it is considered a high energy compound by virtue of containing all the energy which is provided by the 3-phosphoryl group of ATP. Now this S-adenosine methionine is going to act as a methyl donor, which will be utilized for various 1-carbon reactions. So I hope you can recall some of the 1-carbon reactions we have already discussed during the course of other amino acids. So methyl transferase will be the enzyme which will be involved during the various transmethylation reaction, and in this course S-adenosine methionine will itself get converted to S-adenosine homocysteine. Now the examples of transmethylation you must know, because that is important for your viva also, and it can come as a short note also. What is the importance of S-adenosine methionine, how it is being synthesized, what are the various examples of transmethylation reaction. So if you can recall, we have already discussed about the conversion of norepinephrine to epinephrine, how epinephrine gets converted to metanephrine, and also the synthesis of serotonin from tryptophan. So we have already discussed this transmethylation reaction in which S-adenosine methionine is acting as a methyl donor. Then a number of other examples are there which involve synthesis of creatine, which is again a high energy compound. Then formation of methylated tRNA bases, and also methylation of various proteins which will be utilized for the synthesis of proteins. So this is the first role of methionine, transmethylation reaction in the form of S-adenosine methionine. Now by this transmethylation reaction, S-adenosine homocysteine is going to be formed. So what is going to be its fate, we are going to discuss. But then what is the significance of this transmethylation? Apart from making many functionally active compound, it also helps in controlling the turnover rate of protein. And it is said that methylation protects, once a protein is methylated, it is protected from degradation. And also in plants, this S-adenosine methionine, it helps in ripening of the fruits. So this is the significance of transmethylation reaction. So once S-adenosine homocysteine has been formed by transmethylation, it will undergo degradation by hydrolysis. It will undergo hydrolysis in the presence of enzyme adenosine homocysteine hydrolase, which would release adenosine and convert it to homocysteine. So in this form, free homocysteine is going to be formed. So now what is going to happen to this homocysteine which has been formed? This homocysteine can also further have two types of fate. This homocysteine can again lead to resynthesis of methionine, thus completing the methionine cycle, where methionine forms adenosine homocysteine leading to synthesis of homocysteine, and then homocysteine in the presence of enzyme methionine synthase, it will form methionine, and the methylation of homocysteine, it will require two vitamins, the involvement of folic acid in the form of tetrahydrofolate and vitamin B12 in the form of methylcobalamin. So these two vitamins will be required to complete the synthesis of methionine from homocysteine. So you can see how nature is preserving, because methionine is an essential amino acid. So that's why it is being utilized in the body to the maximum. So this is the first fate. Now, but if we have sufficient stores of methionine and we need not synthesize it again from homocysteine, then homocysteine may have another fate. This homocysteine, with the help of serine, it can be utilized for the synthesis of the other sulfur containing amino acid, that is cysteine. Now here again the importance of vitamins is important. Pyridoxal phosphate will be required here again as a coenzyme. So this will be a two-step process. First, homocysteine will react with serine leading to synthesis of cystathionine. The enzyme required here is cystathionine beta synthase, and then cystathionine will be converted to cysteine. For both this reaction, pyridoxal phosphate is required, and the second enzyme is cystathionine lyase. Alpha ketoglutarate will be released with the release of cysteine. Now, though you may not need to remember these reactions, but it is important to note that there are defects related to both these enzymes which are utilized for the synthesis of homocysteine to cysteine. So that is why it is important to know the pathway, so that whenever we discuss the disorders, we are able to understand them properly. Now, what is the significance of this homocysteine and why we must know about it? Because it has been seen that high levels of homocysteine, it acts as a cardiovascular risk factor. In young patients, when hypertension is detected, when cardiovascular disease is detected, then a number of investigations are done to find out the reason, and high homocysteine level, it acts as a risk factor for cardiovascular disease, because it has been seen that homocysteine, it promotes oxidative damage, inflammation and dysfunction of the endothelium, and because of its oxidative property, it forms highly reactive intermediates which can react with low density lipoprotein, which form various aggregates which lead to atherosclerosis. You already know about the process of atherosclerosis, how LDL is a bad cholesterol and it can increase the risk of cardiovascular disease. Apart from this, homocysteine, when it is in excess in the body, it will get converted to its dimeric form, homocysteine, which is again relatively less soluble, poor soluble in plasma, and the damage to the endothelial membrane is again more. So homocysteine is a risk factor for cardiovascular disease. So its level can be measured in the blood to see whether a person is having high levels of homocysteine or normal. So now let's briefly discuss this homocystinuria. So this is defined as a group of metabolic disorders which are characterized by accumulation and increased excretion of homocysteine in urine. So there will be increased presence of homocysteine in the blood, as well as excessive excretion in the urine will be seen. Also, acetylenosin methionine which is excreted will be high, and high levels of methionine will also be noted. So though different type of enzyme deficiencies are responsible for this, the classical homocystinuria, that is because of the metabolic defect in the metabolism of homocysteine, while the other types, the type 2 to 4 which are defined, they are defect of remethylation of homocysteine to methionine, and then they are also mostly because of the defect of the vitamin enzymes, the various vitamin coenzymes. Their metabolism is responsible for the disorders of other type of homocystinuria apart from type 1 homocystinuria. So type 1 or classical homocystinuria, which is an autosomal recessive disorder which occurs because of the deficiency of enzyme cystathionine synthase. So this enzyme is responsible for combining homocysteine and serine, and cystathionine has to be formed. But if this enzyme is deficient, the reaction is stopped here and accumulation of homocysteine takes place in the body. So homocysteine is predisposed to thrombosis, and defect in the collagen synthesis is also seen because homocysteine interferes with the cross-linking or the normal architecture of collagen fibers. So what are the clinical features? Mental retardation, skeletal deformities because of the defect in the collagen synthesis, osteoporosis, and dislocation of eye lens. That is one of the classical finding of this homocystinuria which will be seen. The eye lens will not be at its proper location but will be dislocated because of the collagen abnormality. So apart from atherosclerosis, other features will also be seen, and it can be a cause of death in the early age because of either myocardial infarction, pulmonary embolism, or stroke. So this is a picture showing the abnormal position of the lens in homocystinuria. Now how the patient can be managed? Some of the patients, because basic act as a coenzyme for this particular type reaction, which is enzyme for which is deficient here. So some patient may respond to pyridoxine, although folic acid supplementation has also been successful. And the other dietary modification can be diet which is low in methionine, but you have to provide cysteine so that the sulfur in the body can be of sufficient amount. And then type 2 to type 4 homocystinuria, which occur mainly because of the defect in metabolism of folic acid and cobalamin. The other disorder which is related to the metabolism of this methionine is cystathioninuriia. So here the enzyme cystathionin lyase is deficient, and there occurs accumulation of cystathionin in blood and urine, which is normally not detectable, but because of the deficiency of this enzyme, their levels increase in the body, and mental retardation is the important feature which is seen here. And again, vitamin B6 tends to help in few cases. So this was about the metabolism of methionine, which is involved in transmethylation reaction as well as synthesis of cysteine, and homocystinuria is the clinical disorder, most important clinical disorder which is associated with it. Now we move on to the metabolism of the other sulfur containing amino acid, cysteine, which you know is non-essential, and it is again a glucogenic amino acid. And cysteine plays an important role in the synthesis of various body proteins, so that is why it has to be present in the body. And or apart from that, It is an important amino acid which will be present at the catalytic site of various enzymes, and other compounds which are synthesized from it include coenzyme A, glutathione S, and taurine. So this is in nutshell the metabolic fate of cysteine. Cysteine will be synthesized in the body from methionine, and serine will be utilized for the synthesis of various body proteins. Glutathione, which is a tripeptide, we have discussed it a number of times, will be utilized for the synthesis of coenzyme A along with pantothenic acid. Then it will be utilized for taurine synthesis, and being glucogenic, it is producing pyruvate and also sulfate. Now this sulfate will be utilized for the synthesis of the active sulfate molecule, which I am going to discuss in the next slide. So this is how the cysteine which is being produced in the body is going to be utilized. You have to remember that cysteine in the body can be synthesized from methionine and serine. Now this cysteine can exist in a dimeric form in the form of cysteine, in which disulfide linkage is present. The enzyme which will be catalyzing this reaction is cysteine reductase, and NADH will be required for its this reaction as a coenzyme. Now this cysteine, it can be undergo decarboxylation producing mercaptoethanolamine, which will be utilized for the synthesis of coenzyme A, which is the active form of pantothenic acid. Then it will be the important products, other sulfate will be utilized here. Now this sulfate which is utilized, which is produced by the metabolism of cysteine, with the help of ATP, it can form PAPs. Full form for PAPs is 3'-phosphoadenosine 5'-phosphosulfate. The short form is PAPs, phosphoadenosine phosphosulfate 3'-phosphoadenosine 5'-phosphosulfate, which is activated sulfate, which act as a sulfur donor for a number of reactions in the body, for the synthesis, for the detoxification, for the steroid synthesis, etc. So wherever you will require sulfur in the body, PAPs is going to act as a sulfur donor. And apart from that, it will synthesize taurine, which you know it is required for your secondary bile acids. They will be utilizing this taurine and cysteine. So this is about the metabolism of cysteine. Now let's see what are the disorders which are related to the metabolism of cysteine. Important point to remember here is PAPs, what is the activated sulfate molecule, how it is being formed, and then the disorders. So the first disorder related to cysteine metabolism is cystinuria. So as the name indicates, there will be excessive excretion of cysteine in the urine. So this is a defect in the membrane transport system, where the transport mechanism related to the transport of cysteine and various basic amino acid is defective. Not very uncommon, prevalence is 1 in around 7,000, and there is, along with the increase of cysteine and cysteine excretion, there is excretion of ornithine, arginine and lysine also increases. So overall it is called as COL, standing for cystinuria, ornithinuria, argininuria and lysinuria. So as we have seen that cysteine can be converted to cysteine. So it precipitates whenever excess of cysteine is there, it gets converted to cysteine. So it will, being more insoluble, it will start precipitating in the kidney as well as urinary tract, producing various renal stones. And these renal stones which are having cysteine in them, they are seen as hexagonal crystals when you do the microscopic examination of the urine. So this is a picture, of microscopic picture of a cysteine stone which is seen in a patient of cystinuria. So the patient has to be managed by taking low cysteine diet and increase the fluid intake, because in all type of renal stones, increasing the fluid intake may help by flushing out the excess of substance in the body. So So cystinuria is one disorder related to the cystine metabolism. Then the other disorder is cystinosis, which is also called a cystine storage disease. So this is a type of lysosomal storage disorder where deposition of the cystine crystals takes place in various body tissues, which include kidneys, bone marrow, cornea, conjunctiva, reticuloendothelial system, etc. So a large number of body tissues are affected because of the accumulation of cystine, though the exact cause is not known. One postulation is, as I told, it is a lysosomal storage defect. Lysosomal function is compromised here, and the other is there is some defect in the transport mechanism for cystine, which is responsible for this disorder. So, but this is a dangerous condition which can lead to death in early life, compromising the function of a number of body tissues. Kidneys here are the most important, mostly affected organ. There will be generalized amino aciduria as well as excretion because of the dysfunction of the kidneys. Glycosuria will be there, as well as abnormality of the kidney. It can further lead to rickets and osteomalacia because of the excessive excretion, defect in the vitamin D and calcium metabolism. And renal failure can take place at an early age, and by the first decade the person may die because of this cystinosis. So this is about the cystine metabolism. So this is about the summary of the sulfur containing amino acid. The carry home points are in metabolism of methionine, you must remember what is transmethylation, how S-adenosine methionine is being synthesized, how it is being utilized, what is the significance of homocysteine, okay, and what are homocystinureas, and in cystine, the cystinuria, cystinosis, as well as what is the active form of sulfate and how what is active form of sulfate. This is a common question in the Viva. So that's all about the sulfur containing amino acid. If you have any doubts, you can ask me. Thank you.